Good afternoon, everyone. I am Rebecca Rodriguez. I'm the Central Repository Director, and I want to welcome you all to this webinar. This is our July monthly installment. And as you may already be familiar, this webinar series is part of our Central Repository's broader data-centric efforts, and it serves to prepare our research community for future data challenges and educate the next generation of data scientists. As a reminder, these sessions are held on the last Thursday of every month. And today's session is about some of the highlights on real world data. We're going to have some experts today. This session is geared towards researchers interested in leveraging real world data in biomedical and behavioral research. And we're going to have our experts today are going to be Jasmine Pua and Haitham Siddiqui. Jasmine is head of government solutions at DataBand, leading data strategy, enterprise, and architecture, and data governance for public sector clients. Jasmine previously co-founded a Health Data Link, which is a privacy-preserving record linkage technology company, and now is part of DataBand. Prior to that, Jasmine was the executive director for a public-private partnership with the state of Illinois Department of Health, serving as a health data hub for the region, connecting data for hospitals, labs, and acute care facilities, and other skilled nursing facilities throughout the state. Dr. Siddiqui, he is the technical lead for the National Clinical Cohort Collaborative, N3C, at the National Cancer for Advancing Translational Science, NCATS, and his experience spans the public and private sectors, where he has directed the development of cutting-edge AI solutions for real-world evidence, biomedical imaging, and clinical data interoperability. He has developed and implemented novel machine learning models for causal inference, patient phenotyping, and early detection of sepsis. He also did the graph deep learning solutions to analyze healthcare provider networks, and he holds a PhD in biomolecular engineering and a master's degree in applied mathematics from the University of Notre Dame. I will give it to the experts now. Thank you.
Thank you so much. Can everyone hear me? Great. So the session goal for today is to do a super quick whirlwind tour of types of real world data, talk a little bit about what the potential of real world data uses can bring in terms of use cases and considerations. From there, I will be handing off to my colleague Haitham to talk about a real world use case where the N3C is a privacy preserving real world data research infrastructure. There's lots of different considerations as you think about constructing these sorts of infrastructure, and we hope to be able to answer your questions. Just for everyone to know, I cannot see the screen, so if you have questions, I will get to them at the end of the session. Great. So what is real world data? Our definition of real world data is twofold. One is it is commonly accepted as information that's collected through routine healthcare activities. But as you all know, both social and commercial determinants of health greatly impact health. And so, when we're thinking about real world data, there are traditional sources, so electronic health records, your labs, your pharmacy data, but we also think about social and behavioral data, even financial data, other kinds of things that really show us the picture of what's going on in your life because it's very material to how you're able to cope with healthcare delivery, treatment plans, all sorts of considerations when it's part of your treatment journey. In terms of why we think this is important and how it's related to the prior webinar, which covered linkages. As you all know, healthcare is incredibly fragmented everywhere, especially in the US. And so if we take an illustrative patient here, you can see all the different events in someone's life with just even changes in location of where you live. If you layer in the complexity of trying to get your electronic health records across your different physicians and care teams for triage, and we think about this longitudinally from birth to death, the picture of how you assemble the patient 360 starts to get really complex. And so there's kind of two ways that we think about this. There's readily available data, and then certainly there's primary data collection. And we'll be talking a little bit about this more. But, as you look at this picture and you think about assembling this for cohorts of interest, or just broad populations, especially for things like common chronic diseases in particular, the picture starts to get really, really complex. How can we construct a methodology here to kind of bring together all this data and use it? One thing that would love to have you all be thinking about is really to rethink the use of data. And a large part of this is about data reuse. As you know, obviously here within NIH, as we all conduct our research, data reuse and kind of like the FAIR data principles are incredibly important. And we do think of real world data as just one tool as part of your methodology as you're thinking about constructing studies. So while I won't go into all the different use cases, we should think about what sorts of data are readily available that can be repurposed to enrich or augment data sets that you might be looking at. But there's also kind of third-party data sources that you might bring into the mix as part of your study design. So I've listed a couple here to consider. So for example, if you're using multiple data sets to create a single cohort, that's certainly one version of it. What we have seen, and this is true as well within real world data and real world evidence work that we've started to see within FDA, for drug submissions, is construction of comparator arms, particularly when we are unable to go back and randomize individuals in a way that's ethical within an RCT framework, right? So please do start to think about real world data in terms of hybrid approaches, because again, like it's one more tool, and part of it is lowering the patient burden in terms of primary data collection. I'm going to run through just like one additional example beyond this. But for example, when we're thinking about constructing a study, obviously there's kind of like, how do you go back for your cohort of interest and get their retrospective natural history? So that's one very tangible use to really understand the patient journey that led the patient to like the enrolled study. The other part of it is obviously after the study, so prospective data collection. We tend to think of this more as long-term follow-up or post-market surveillance use cases in particular, but we do think of these as like passive data collection. So it's a situation where, as you all know, with cell and gene therapies, more and more coming to market, the timeline to be able to follow into the future prospectively also increases. It's a huge burden, but there has to be a way to continue to monitor 10, 15 years post-therapy. And then finally, I think this is the part that tends to get missed, which is as you're running your study, we're all trained to perform analysis at the end of the protocol, after everything is completed. But what we have seen is the incorporation of kind of a concurrent model where data is actually used for enrichment. So you can understand population, whether there are subcategories or subcohorts that are starting to emerge that might actually affect your study or potentially be confounders for your study. And part of that is being able to incorporate kind of set additional data for enrichment during the study itself. So that's how we think about the spectrum of how real world data can be used throughout the study. Another use case that's very, very different that is now possible with the reuse of data. This was a use case here where I'm based in Chicago. We have a community-based engagement model here where pastors were P Corp, pastors were trained, IRB qualified, they know how to administer studies. And part of that is being able to reach at-risk and hard-to-reach patients where they are in the community. So if you want to, if that is the target population for intervention, if that's the population that is not currently being served, the best way is to construct novel methods where you can reach them where they're at. Now, the challenge with that, as you all know, is then there's a very long chain of potential screening and eligibility. So this is a circumstance where the Chicago Area Clinical Research Network basically pre-ran the phenotype for a study. And this was based on linked deduplicated data throughout Chicago institutions. And what would happen then is the community-based organizations would engage with the potential participants, discuss with them, collect basic information to generate kind of de-identified tokens. Tokens are used to link data in a de-identified way. And so this was a really quick and easy way to really screen out individuals who didn't meet the inclusion and exclusion criteria without the community organizations needing to basically burden the patient with a very, very lengthy screening protocol. So we highly urge that you think about your research and study designs in a way that incorporates this sort of data in novel ways. I will now hand it off to my colleague Haitham.
Thank you, Jazz. I'm going to go ahead and share my screen here. All right. Hopefully you all can see my screen.
Yes.
All right, fantastic. Thank you. So I wanted to pick up where Jazz left off and talk about the National Clinical Cohort Collaborative, or N3C, really in the context and the framework that Jazz really laid down for us, talk about where it began, where it is, and where we're headed, and how it relates to a lot of the challenges we face when working and dealing with real-world data. So really, what was the impetus behind N3C? So N3C emerged during the pandemic as a way to solve a problem. And that problem was that, given kind of the unprecedented situation, there was an urgent need for observational data at scale. And as Jazz really demonstrated with her, patient journey or patient timeline, here in the United States, there's really no centralized healthcare. And as a result, there's no centralized healthcare data. And that applies at many different levels. And that can be at the level of an individual whose healthcare journey is split up across a multitude of different providers and institutions and data sources. But it also means that the nature of the data that's stored at these different institutions and organizations are different. And so you kind of face multiple challenges in being able to reconstruct a patient's journey to be able to tackle public health problems. And so with these things in mind, NCATS, in partnership with the CTSAs, CTRs, OCHIN, and a variety of other agencies, sought out to build N3C as a solution to this problem. Now, before I continue, I do want to point out that there are really two, broadly speaking, two paradigms when it comes to large-scale observational research. One is a federated approach and one is a centralized approach. There are pros and cons to each model, but with N3C, we adopted a centralized approach, which is to say that we aggregate and bring the data into one location and make that data accessible to investigators. And the advantage of adopting a model like that is you do allow more flexibility in terms of the kinds of scientific questions that you can ask of these data. Now, N3C did begin during the pandemic, and ultimately, the way we see that is we saw that as a powerful proof of concept, right? So it really demonstrated the value of large-scale clinical data harmonization, collaboration, and NCATS and its partners made significant investments in building this robust platform, and I'll talk about that in more detail here. And so we're very interested and we're actively working to expand beyond COVID-19 into chronic diseases and complex conditions and other areas like that. And we also really care about education and bringing what we've built in N3C to other communities, such as ourselves and other individuals who may not have experience with N3C. And so also just to kind of set the stage for the context of the scale we're dealing with N3C, because one of the things that you kind of run into again and again is that you face new engineering challenges, new scientific challenges when you try to scale, right? So sometimes when you build a pilot, something that's small, things work very well, but the moment you start scaling that model to try to operate at a much broader level, you run into new challenges. And so I really wanted to drive home the scale we're talking about when it comes to N3C, and then we can kind of jump into how we dealt with some of those challenges from a data and infrastructure perspective. So N3C was launched in 2020, and by 2025, so today, 5 years later, there have been over 5,000 citations of N3C driven research. If N3C was a researcher or investigator, it would have an H-index of 33. Over 1,500 authors have participated in the N3C collaborative, and it's really been transformative across the board for all the areas where N3C has been active and contributes. But as I said, as we transitioned away from our focus away from the pandemic and more broadly trying to capture other disease areas, we had a phase 1 clinical pilot, and we are now in a phase 2 clinical pilot. And the idea there is to take that model and to try to apply it to other disease areas and domains. Initially, we started out with Alzheimer's, COPD, and renal disease. We had 12 institutions that participated in that phase 1 pilot. And now in our phase 2 pilot, we're focusing more on better data linkage, on data integration, and on supporting new tenants like cancer and renal, and new partnerships as well. We've had continued partner community engagement. So we have 96 institutions that signed our data transfer agreement. So institutions from across all 50 states, DC and Puerto Rico. So that's the kind of extent of the nature of data that's being aggregated into N3C. And I did talk about the ongoing pilots already. All right. So with all that context set, how does N3C work? So at a very high level, the way N3C works is we have partnering institutions that contribute data into N3C. I did mention it's a centralized model, so it's a centralized enclave. And because different institutions, they typically adopt different, what we call common data models. So there are different ways of structuring EHR data. We have to build pipelines to maximize the amount of data that we can aggregate to maximize utility for investigators. We have to be able to accommodate a variety of different common data models. And so this is where we take data in, whether it's OMOP or CORNET or ACT or Trinetics or I2V2, and we bring these data in and we harmonize them, and then we make them available in the N3C data enclave. So it is a secure data enclave. There's a very sophisticated governance structure in place and a set of access controls. But it is ultimately a public resource, and that means that investigators who go through the appropriate qualifications and compliance checks are able to gain access to these data and carry out research on this data set. And so the underlying foundation to our system is OMOP. That's a common data model. That's what we could call it like an analytic data model. So it's designed for observational research. And it's backed by Apache Spark. And so that enables investigators to leverage everything from SQL to Python to R to carry out their analytics within the N3C data enclave. So really jumping into the structure of our data governance and access. And I would say there are 3 key components to how this is structured in N3C. And the 3 primary stakeholders are the data users. So this is the research community. the investigators that want to come in and leverage this resource. Then there are the data stewards, that's NCATS. So we do not own an institution's data. We are simply the stewards of that data. And then there are the data contributors, the institutions that we partner with that ultimately contribute the primary data sets that make up the foundation of the data that's available in N3C. And so the way this works is N3C and NCATS sits at the interface of both the investigators, the research community, and the data contributors. In order for institutions to be able to contribute data, they have to have the appropriate IRB in place, and then they have to have the appropriate data transfer agreements in place. So that's kind of the data governance aspect to be able to contribute data to N3C. What we request from institutions is what we call, it's a HIPAA limited data set. And so that data set has the HIPAA direct identifiers removed with the exception of dates of service and 5-digit zip codes. And so that payload is transferred into N3C. And then that data is harmonized and curated. It goes through rigorous data quality checks, and it is integrated into one large data set. And then on the other side, when you have an investigator or a data user who wants to come in and leverage these data in N3C, the first thing they have to do is they have to ensure that their institution executes what's called a data use or data user agreement with NCATS. They have to register. They have to attest to abiding by our code of conduct and our community guiding principles. They have to complete the appropriate human subjects training and IT security training. And then they can enter into N3C and then submit what we call a data use request to get access to data. And that ultimately is where they specify the specifics of their scientific questions that they want to ask and the justification for their questions. And we also have a tiered data access model. So investigators can get access to synthetic data. So that's the easiest data set to get access to. Then we have the identified data and then that limited data set that I just talked about. And so the primary data set that investigators would get access to for research would be the de-identified data set. Investigators can also access the limited data set if they have a letter of determination from their institutional IRB, but they also must provide a research justification for minimum and necessary. So why is it that the de-identified data is insufficient for them to be able to conduct their research and answer the specific questions of interest to them in N3C? And so this is kind of the governance model, which really sets the stage for how we ensure we are kind of respecting the privacy of the patients whose data we make available and abiding by the agreements that we have in place with the institutions that very generously partner with us and contribute their data into N3C. So this model worked very well during the pandemic, and when we wanted to kind of expand into other clinical domains, we had to reassess specific things. So really the key thing to emphasize here is this original limited data set that the institution is sending is ultimately bound by a phenotype. And so phenotype here really just means a set of patient inclusion criteria. And for the purposes of COVID, they were a set of COVID positive patients and a corresponding set of demographically matched controls. And while these patients were suitable for COVID-related research, they are not necessarily suitable for other research in other clinical domains. And so the idea is, well, not only do we want to expand into other clinical domains, but we also want to set up a model that can scale well. And so what is suboptimal, of course, is for us to have to go back and renegotiate or re-execute agreements with institutions each time we want to expand into a new clinical domain. So in this diagram here, I have, for example, a cancer tenant. So this is where cancer-related research would occur, and then a renal tenant where renal-related research would occur. And so the idea is that we have a single parental data transfer agreement that we would enter into with an institution, where that institution would contribute what we would call a disease-agnostic phenotype. So this would capture clinically meaningful patients at an institution. Roughly speaking, that is patients with 2 visits at least 30 days apart. So we look for some level of continuity of care at an institution. And then these data, they enter into N3C in a holding tank, and it's locked down. And that data is then released into individual tenants based off of a disease-specific phenotype in partnership with the data contributing institution. And so they would say, we want to participate in renal or in cancer. And then the data would be released into each of those two tenants. And then the investigators would be able to establish projects within each of these tenants to carry out domain-specific research. The other thing that you can see on here is I have this PPRL commercial data registry data section. I'm going to talk a little bit more about that, but we do data linkage in N3C, and there are lots of good reasons for us to do that as well. So really, the objectives of N3C Clinical, and I know speaking to an audience and NTDK here, is ultimately to, NCATS, we were trying to understand the financial and infrastructure and community resources needed to develop and maintain these tenants. And really, it's around refining operations and governance and technical architecture. But equally important was for us to be able to establish, and I would say, transformational partnerships with a variety of agencies, and that includes CMS and HRSA, NCI, and of course NIDDK and other HHS agencies. It also gave us an opportunity to explore next generation healthcare interoperability. And so we've been working on incorporating support for FHIR US Core, and that will allow us to kind of expand the remit of N3C to think about health information exchanges and being US CDI compliant and engaging with other regulatory and compliance expectations that are kind of both in place with the Cures Act and also coming down the pike in the future. And really establishing a set of new capabilities that will expand the space of the kinds of scientific questions that can both be asked and answered by a resource like N3C. But really, all of this discussion is limited in impact if we restrict ourselves exclusively and solely to electronic health records. And really, the reality is electronic health records are not enough. And I think Jaz talked about this in terms of the nature of fragmentation. And so really, why is it important to have multiple data sources? Because each one data source just gives you an incomplete, kind of a partial snapshot of the overall patient. And so by linking in multiple data sources, EHR and other, you're able to overcome really the limitations of these siloed data sets. You get a more complete picture, and you get greater continuity, both longitudinally and kind of horizontally on a patient. And really thinking about it from a scientific angle, you reduce your bias in observational studies. So you have a more accurate measurement of exposures and covariates and endpoints and all the things that matter for the kinds of research that investigators conduct. It also enables kind of more foundational research or investigations, which is to look at cross-validation, right? And so there are some challenges that are associated with multiple data sources in that they can sometimes contradict one another. It's very nice when they're all complementary, but sometimes they can contradict one another. And so carrying out cross-validation when you encounter either conflicting or complementary values can enable you to reconcile data, enhance data integrity, and inform the research communities writ large as to the advantages and disadvantages and the limitations of the various data sets and how they affect research as well. And so thinking through what are multiple data sources in the context of N3C, the other component when you start linking data to think about is privacy. And so we leverage a framework called privacy preserving record linkage. And so the idea behind that is you are connecting records using a secure pseudonymization process in a data set in a way that you can refer to the same individual across different data sources while maintaining the individual's privacy. I know that's a mouthful. I have a nice picture that'll illustrate what that looks like. But what I wanted to do is talk a little bit about data sets that we bring into N3C. One very big one is CMS data. So I think for all the reasons that we can all imagine, CMS data is very important. And there are specific populations of patients where CMS is a primary or the primary data source for studying them in an observational context. On top of that, there are social determinants of health that can be linked and integrated with patient data to understand environmental exposures and other factors that may affect patients' healthcare outcomes. During the pandemic, we brought viral variant data in to enable people to really differentiate and study the COVID-19 exposure at the level of the different variants. One other very important data set that we brought in and we maintain across our COVID and clinical domains is mortality data. So if a patient is discharged from a hospital and they happen to die elsewhere, making sure that we capture that loss of life is very important in a lot of survival analysis and a variety of different investigations. And so having a robust mortality data set with very good coverage, so thinking like National Death Index, but also with very up-to-date coverage. And so being able to aggregate data from private and commercial sources to give us a more complete and up-to-date picture at the level of weeks, perhaps, is also very important for very time-sensitive research. And then, of course, we have the foundational EHR clinical data that's present. We had brought in vaccine data at a time, and then imaging data for MIDRC. So radiology and a whole host of other kinds of medical imaging are very important for specific research. And one that I'm very excited about is to be able to bring in registry data. So with NCI, the National Cancer Institute, we partnered with them to be able to bring in the SEER Cancer Registry and the NACER Cancer Registry data from the institutions contributing data. We also partnered with NIDDK and with HRSA to be able to bring in the transplant registry to focus on kidney transplants for the renal tenant. And we really expect that being able to link these data sources alongside the EHR data will open up new avenues of research that otherwise have not really been probed very thoroughly due to the really inability to bring these data sets all in one location and to be able to study them that way. And so very briefly, I wanted to just cover how linked data flow happens within N3C. So, I did talk about different data sources, and so the details can be a little different depending on them. But very broadly speaking, there are the N3C clinical sites that are contributing the EHR data to N3C. And so what happens is they do what's called tokenization of the data. So we leverage Datavant tokens in our system. And so those tokens leverage specific identifiers that exist with the clinical site to generate hashes that are non-reversible. And then those hashes, those tokens, get sent to what we call a linkage honest broker. So they stay at arm's length away from any patient data, and they just do the matching with those tokens. And then whatever other external data source we want to bring into N3C, that same tokenization process happens. So the linkage honest broker, they match up the different tokens across the different data sources, and they hand off what we call like a crosswalk or a linkage map, linking these hashes to one another. And then that hash map goes into N3C, and within N3C, then we're able to connect the mortality data with the EHR data, the data for a patient who went to Duke and then UNC together, but we don't know the identity of that patient, and all of the above, really to connect these records across the different data sources for deduplication, for enrichment, and for all those other reasons as well. And so then the question arises, I think we all intuitively understand the scientific need for data linkage, but how important is it really? And so we carried out a number of investigations looking at the scientific impact or the validity of specific scientific investigations based off of linked data versus non-linked data. And so one example I wanted to present here is we were looking at the risk of stroke post-vaccination, and we leveraged a number of different methodologies. One was target trial emulation. The other one was self-controlled case series. And we said, if we went into this investigation and we used EHR data alone, or we used claims data alone, in this case it was CMS, versus if we used EHR and claims data together, how would this affect the specific outcome and effects estimate of interest? And what you see here, and so this is a very well-studied problem. So we know that there is a no association between this particular vaccination and stroke, and so we expect there to be no effect. And so what we see with the claims data is we get a significantly a biased result that is significant. With the EHR data, we get a non-significant result, but it has greater bias and larger variance. And it's really when we combine these two data sets together that variance shrinks and the overall point estimate approaches unity, which is what we would expect. And so we do see real tangible impacts of data linkage on the kinds of the potential scientific validity of the questions that investigators ask. Of course, the reality is your mileage may vary depending on how sensitive the research question you're asking is to the different data sources. And so, of course, we conducted a variety of sensitivity analyses and ablation studies. But this isn't kind of the scope of what I want to highlight. But I hope the point I'm trying to drive home is the importance of leveraging multiple data sets in your scientific investigations whenever possible. And really here, I also wanted to transition and talk a little bit about our partnership with NIDDK and talk about some of the goals of the renal tenet that we were trying to establish and we are establishing is we're trying to really better understand and conceptualize patient journeys through CKD and dialysis and transplant and beyond, and really understand the differences in outcomes or death rates across data sets, understand transplant referrals and organ allocation and bias, and quantify all kinds of issues in underserved populations, and actually be able to get at the heart of answering some very nuanced questions that are only possible when you combine EHR, billing, and transplant data. So that's the CMS data, the EHR data, the SRTR data together. And really, without the ability to combine these things, it's very difficult to arrive at the correct results or conclusions. And of course, we want to create an open science community and foster and facilitate team science as well. Really, linked data and good algorithmic practice is at the heart of investigations like these within the renal tenet. And we're within kind of the things that are priority for HHS. Identifying the added value of linking data sets in the renal tenet is very important. And so this is where we kind of have a marriage of those three primary data sources, EHR, CMS, and SRTR coming together. And our goal really is for this to be a testbed for validation of machine learning and AI methods as well applied to these patient journeys that I talked about. And so I just wanted to close out by talking a little bit about the future of N3C. So the model I highlighted where we have these specific, these disease-specific tenets is very valuable and very powerful. But really, as we think about scalability and growing beyond specific disease domains, or in thinking about areas where different disease domains intersect, so thinking about renal cancers or thinking about the impact of, or the likelihood of diabetic patients developing other conditions that might not be within diabetes or pulmonary, et cetera. Having a model where we have these distinct isolated tenets can be somewhat limiting. And so what we're exploring right now is the ability to dynamically provision workspaces with an investigator-provided phenotype. So an investigator would say, I really want to study this very specific population, these very specific controls. And what we do is we ultimately provision a project or a workspace very specifically with the patients that are absolutely minimum and necessary to answer that specific scientific question. And so we are really aligning data access with scientific intent and opening up the ability to be able to answer research questions that transcend or that cut across multiple clinical or disease domains. And really what I wanted to close out with is to kind of think about from the perspective of the research community and investigators and investigators at agencies thinking very broadly is what, with this framework that we lay out for N3C, what is our hope for how things can be different in this model? And so the example I give here is, imagine you are want to study a specific scientific question, in this case, sepsis, for example, with 5 other institutions. Between the administrative setup that's necessary, the legal negotiations that have to happen, establishing the technical infrastructure, building a platform and acquiring compute and storage, and then having to harmonize and clean and link data and acquire external data sets to go from ideation to initiation of a project, you are looking at well over a year, oftentimes longer, significant delays, all kinds of novel legal hurdles that you would have to go through again and again for every individual research project or collaborative that you want to establish. Whereas within N3C as a national level resource that brings all these things together, that partners with different agencies to facilitate research across different clinical domains, investigators can bring their question into N3C. They can submit a data access request. They can execute their agreements, and within the span of weeks, they can potentially go from the initial setup and the kickoff to actually having a research environment where they can conduct their research. And so this is kind of the vision that we're hoping for and how we want to facilitate and enable collaborative research at national scale. And so with that, I'll stop here and open for any questions.
Thank you. Excellent presentation. I appreciate both of you. There are some questions in the chat. If you want to address them, I also have some additional questions, but we can first address those that are in the chat.
Yeah, I see a question here. I'm going to read out for folks. What are key considerations and best practices researchers should keep in mind when generating, stewarding, and seeking to obtain and use secondary data from electronic health records and real-world data sources, particularly in terms of patient privacy and data security? That's a fantastic question. I think, as you all know, 80% of all our time is not on analysis. It's really on data governance. So as you think about real-world data sources and data originators in particular, I think that is a major consideration. I'll give you two examples. Many commercial data sources tend to accept one standard for data de-identification, and that's de-identification based on the HIPAA expert determination standard, and that's in the HIPAA privacy rule. What that means is a statistical analysis and assessment needs to be conducted on all the variables you're seeking to analyze as endpoint collectively. So it's very much about a fit for purpose assessment of the risk of data. So if you were to use or enrich your data with, let's say, a commercial claims data source, then from a data governance perspective, you have to allow time, process, that negotiation of endpoints and obfuscating or extracting or abstracting variables so that it doesn't increase re-identification risk. So that's one version. To Hytham's point, as he kind of like talked through the data transfer agreements, the other method is to construct a different model from scratch. So for example, with N3C, health system sites, FQHCs, have all signed a data transfer agreement. Now, when that was first constructed, we conceptualized of it as a limited data sharing network. That's very, very different because it does not necessarily have to run through the HIPAA expert determination method. And so again, it falls under common rule as well, right? It gives you more flexibility. So those are two examples of like considerations you should keep in mind. Obviously, there are trials and studies where you're enlisting participants. For those, what we recommend is making sure that data linkages are in the informed consent and they are explicit. One thing we've seen in particular with registries is folks will come to us, they're like, yes, we want to do these data enrichment studies. We've got 5 versions of our registry agreements with health systems over the last 20 years. What is linkable? When we look at those, oftentimes what we tend to see, whether it's in patient level consents or in registry consents of institutions, is the thinking about data linkages tends to be thought of as an analysis activity as opposed to a contractual activity or a consent activity. So we would highly encourage kind of thinking about your study design and your data resources holistically, because if you don't establish the kind of like governance structure from the outset, then, when new technologies emerge that permit data linkages, for example, you're unable to go back to that population. So we have templates that we tend to provide to make sure that if you have informed consent, you address it and address it very explicitly. The All of Us research program does a really good job of making data linkages explicit in its consents. And obviously, it's really important to understand how those consents themselves intersect with regulatory requirements, right? So highly encourage working with your various Office of Science and Policies, because they are the heart of making this happen. Hytham, do you want to take the next question?
Yeah. Yes. So the question is, given your partnerships, how do you view N3C's efforts intersecting with the new standards announced by CMS for EHR? Will you be able to do more on the EHR front as this develops? That is a fantastic question and very, fresh off the press one. So I would say absolutely. This is very much in line with where we see N3C headed. So we had a very practical consideration, of course, when N3C began in 2020, which is that we want to be able to rapidly onboard as many partnering institutions as possible. And those institutions had a variety of different ways of representing their data and common data models and things like that. But a more principled approach is really to really focus or invest heavily in interoperability, and that is thinking about US CDI and thinking about FHIR US core standards. And so I think I mentioned it very briefly, but we have active ongoing projects with a couple of health information exchanges around really leveraging FHIR US core to enable data transfer really at the level of the kinds of patient populations we deal with. Of course, FHIR is fantastic for interoperability, and the CMS announcement and their specific, I think there's also another page that specifically talks about their interoperability plan and vision, aligns very well with the direction we're headed. And we hope as more institutions are able to rapidly kind of bring themselves in line with CMS's guidelines, I think it'll make our job on the N3C side easier because we have to do a lot of work in terms of harmonizing and curating and handling data that comes from institutions that are very heterogeneous. And so we're very excited about this, and we think it'll make it easier actually for us to integrate with more EHR systems in a less onerous way, both on us and on the teams on the ground at our partner institutions.
Yeah, and really quick to tack on that, Datavant was one of the companies that was part of that announcement. One of the priorities that we have is really access to patient data. So you'll see kind of mention of Blue Button. What we see right now is an incredibly manual process of getting patient records. Obviously, the raw record itself, untransformed, is the gold standard itself. We frequently see requests for that. That is unfortunately not necessarily complete, via the various kind of digital methods today. And so part of this will be working to make sure that, patients themselves have patient-mediated exchanges that are possible and digital. And also for research use cases, there's greater access to this data.
And Jasmine, I have a follow-up question based, following from prior comments and your prior responses. Sometimes EHR and real world data have a bad rep, right? In terms of data quality. How can researchers effectively address and navigate the potential challenges and limitations associated with secondary use of these type of data and the issues with quality, completeness, interoperability? How do you address those things? And especially when then on top of that, you trying to add, or link the CMS data.
Yeah, that's a fantastic question. So I can answer from our perspective, and I'm sure Jazz can have a few thoughts to add there. So I would say we invest heavily in two key areas. One is in resources for education, and also in resources for data infrastructure. So education means for investigators coming in, whether they're new or more seasoned, but are used to working with different kinds of data, maybe closed claims data, those kinds of things. What we do is we provide opportunities for them to attend classes. So we actually have live classes that they can come and attend, where we train them to how to work with these data and identify issues that are present in these data that may not be present in other data sets, and all kinds of things really in that space. On the sort of like a broader data, we also offer like help desk hours, and we make informaticists and epidemiologists and biostatisticians available for people to consult with. But also on the other side, we invest very heavily, both on the institutional side to work very closely with them on their data quality. And so we publish, we don't publish, we provide data quality reports to the different data contributors that tells them, for example, how much missingness and completeness and data density, and we have a very sophisticated and very complex data set of data quality checks, how they compare kind of in an absolute sense and how they relate to other institutions on average, so they understand where they live in terms of like the expectations of where data should be and can be and how things are mapped and those kinds of things. So what we can do is we can mitigate. We also, on the researcher side, provide templates that generate that an investigator can customize for their specific research question that will generate a series of data quality tables and dashboards so they can start investigating things that are specific to them. So they might be interested in a specific set of drugs or a specific set of comorbidities or visits or measurements. And so we provide means for them to understand how these data are available with the different institutions. The fact of the matter is data quality varies from one institution to the next. And depending on the specific question you're answering, one institution may be very good for your question, and one institution may be not so good for your question. And so the idea is to provide as much transparency and to arm the investigators coming in with as many tools as possible to be able to have as much insight into the nature of the data that they are using for their research as possible.
Yeah. And I think there are two considerations as well, because the data is not static, right? In the case of N3C, data is coming in weekly refreshed. What we have seen with various clinical research networks is one tiny change in one health system where the IT team may not have let the informatics or research teams know what's going on will drastically change what's coming in. And so what we've seen are mismappings of data, simple stuff like transposing gender codes, changing of kind of different patient codes, so you don't know which is the unique patient. So it's super important at data ingest to have these data quality processes so you can kind of have a continuous monitoring process. Without that, you don't know when you have constructed the data set where something goes off. Because again, we don't think of data as like a static data set that's constructed, frozen, and that's it. It's super important to understand what went into it, whether it was mapped appropriately. The other method is to characterize the data sets or benchmark it against larger data sets. So, how representative is it? As you all know, as part of a lot of emerging AI research, if your data set's biased, your algorithm is going to be biased. So it's also really important to be able to like anchor and compare your source data set with maybe other reference data sets as well. So those are kind of simple methods. On the linkage side, part of it is when you're trying to reconcile variables that are reported through multiple data sources. So let's take mortality. You could be having mortality reported from government data sources. A family member could call in to report a death at the health system or to a study when there's follow-up. There's EHR reported death. Not all the codes relate to an actual clinically confirmed death, right? Maybe a patient or family member report still shows up as an ICD code. And so when you're thinking about looking at all these different breadcrumbs that lead to the same variable, part of it is also assigning a level of authoritativeness to the data. So if it's a clinician confirmed death, there's a clear encounter and event, that's probably reliable. If you are linking in CMS data, it's a confirmed death, that is probably reliable. If you have third-party data coming in showing that someone passed away within the last week or so, you want to see if maybe there are other activities within the patient's record that demonstrate that the person's still alive. So there are like a lot of different kind of data reconciliation and comparison methods that we have constructed in order to like really ascribe quality at some point before data is released.
Thank you. I cannot underestimate how much importance there is into data quality at the point of data generation and data ingest. So I appreciate both of your times and your thorough responses to our questions. I don't see any other questions in the chat. So Again, thank you. I'm going to take a few minutes to advertise our next session for the month of August. And this is we're going to have some of the users of our repository that have, the impact and innovations that they have been able to do with the use of the NIDDK Central Repository resources. We hope to have speaking Dr. Adam Guerra, Dr. Prasanna Santham, and Dr. Juliet Emmanuel. They will be with us next month, so I hope that all of you stay tuned and join us next month. Thank you again. I think there will be a survey about, giving us some ideas for the next topics. So if you want to all take some minutes to respond, we will appreciate it. Thank you.